Hello. So today we're looking at B1.1, which is carbohydrates and lipids. So I want to start off by posing a question. I'm not going to give out the answer now, but I just want you to think about it and see if you know the answer. If you do, then you might be an expert in this and you might not need this video, but otherwise, I recommend you watch on as I'll give the answer to this question and other questions that I'll give you at the end. So let's get started. What are the main macromolecules of life? So there's many chemical molecules that form part of life, but these are the four main big ones, right? We have carbohydrates, we have lipids, proteins, and nucleic acids. You've probably heard about most of these, but you might actually not know much about them. So that's what we're going to do in this video, right? We're going to focus on carbohydrates and lipids, and then in future sections, we'll look at the other ones. So just in general, this is some important knowledge to just understand everything. What do all macromolecules share? Well, they all have carbon atoms, right? Carbon atoms are great because carbon atoms bind to each other and form covalent bonds. Covalent bonds, in case you don't take chemistry, is a bond where electrons are shared. So two electrons are shared between the two adjacent atoms, and it's the strongest type of bond between atoms. And this allows for very stable molecules to be based on carbon. Now, a carbon molecule can form either four single bonds, as you can see here, right, where it's bound to a hydrogen, another hydrogen, an oxygen, and a carbon, but it can also form double bonds, as you can see here. So in this case, instead of sharing two electrons, you would share two and two, so four electrons. But that means there's no electrons to share with other atoms, right? So in this case, it's only going to bind three things. Similarly, if this carbon had two double bonds to this N and to this O, then it wouldn't be able to bind anything else. So double bonds are stronger, but they also mean that the carbon can bind to less things. But this diversity of bonding means that molecules can adapt very, very complex structures. So as you can see here, rings, right? I've pointed to in the arrow. There's also branches that can develop, and the length they can be very short, but they can also be of a very long length. So this means that we can have a high diversity, right, which is great for organisms because we can get them to do many different functions. So first, how are macromolecules created? Okay, we're talking about really, really big molecules. Again, as I mentioned, there's other molecules, right? For example, we have calcium in our bodies or iron, but those are very, very small. Those are atoms, right? Whereas in this case, we're talking about molecules made out of hundreds or even thousands of atoms. So how are these formed? Well, normally they involve the reaction from monomers to polymers, right? A monomer is one unit. So for example, this glucose is a monomer. And then when you add a lot of those together, you get a polymer, right? A chain, basically. So the chemical reaction, when you link two monomers together, is called a condensation reaction. The important thing about a condensation reaction is that the two molecules end up being linked, as I mentioned, and water is released. This is a vital thing, okay? Water is always released in a condensation reaction. In the end, you'll see that they're bound by an oxygen, right? And if you look at here initially, so basically you lost one OH and an H to give water, and then you got that O that was left over to bind them together. In this case, glucose plus glucose gives maltose, and that's a glycosidic bond, it's called, okay? Also, condensation reactions, it's not shown in the diagram, but they require ATP, very important. I don't know if you know what ATP is yet, but it's basically energy, if you don't know. Great. And then the opposite of that is we can also go from polymers to monomers, right? And that's called a hydrolysis reaction. In this case, we need water, right? Instead of losing water, we need water for the reaction to occur. And then we end up with two separate molecules. So why would we break down polymers, right? Why? Maybe it doesn't make much sense to you. Well, first of all, it's so that we can reuse the monomers to create new polymers in case we need them to do something else, right? We need another function within the cell. Also, it's a source of energy. So when you break these bonds, right? I told you that for a condensation reaction, you need ATP, you need energy. Well, when you do it the other way around, you gain energy. So energy is released, right? And this is what we do during digestion, right? When we eat these polymers, right? Longer than this, of course, but we eat polymers and then we digest them into smaller chunks, into monomers, right? To release energy and then use those monomers to make other stuff. So that's a very simplistic way of explaining it, but I hope it makes sense because this is often misunderstood amongst IB students. So we're going to look at carbohydrates first, right? So starting off with carbohydrates, We have monosaccharides. This is the monomer, right? It's the single unit. It's the simplest type of carbohydrate. So here are two examples, glucose and fructose. You'll see that fructose is a pentose, right? It has 5 sides, right? So 5 carbons. And then glucose is a hexamer, so it has 6 carbons, right? Focus on glucose because this is the one that IB will ask you the most about. So glucose, as I said, is a hexose. It's soluble in water, which is very important. It's a small molecule, which means it's transportable. So the fact that it's soluble and that it's transportable means that it can diffuse in the blood, right? It's stable chemically, which is very important, so that it doesn't just break down spontaneously, because in the body we want to control these molecules, right? And then again, it yields energy. So if you oxidize a glucose molecule, that means you just start cutting off different parts of it. It will give you energy. So this is one of the main ways in which we get energy in our bodies. Perfect. So that's a monomer, and it forms polymers, right? And those are the polysaccharides. So there's 3 polysaccharides: cellulose, starch, and glycogen. Now, cellulose and starch are found in plants, whereas glycogen is found in animals. So that's an important distinction. First, we're going to look at one of the main functions of polysaccharides, which is energy storage. So energy storage is only for starch and glycogen, okay? Cellulose is not, its function is not to store energy. So when we look at energy storage, we have starch and glycogen, as mentioned, right? But then starch can be subdivided. This is where it gets tricky, okay? But I have a summary slide later, so hopefully it'll make sense in the end. So starch can be divided into amylose and amylopectin. Amylose is unbranched, whereas amylopectin is branched. Now, what does this mean? Okay. An unbranched polysaccharide means that the glucose molecules only bind with 1 to 4 bonds, right? That means they bind side by side, you could say, right? Forming a straight line-ish. Whereas when you get branched molecules, that means that they always bind 1 to 4, right? So side by side, but they can also bind 1 to 6, basically on top of each other, right? This 1 to 4 and 1 to 6 refers to the carbon numbers. You can search this if you want more information, but basically 1 to 6 means one on top of the other, 1 to 4 means side by side. And then glycogen is also branched, right? And then finally, these are all made of alpha glucose. So that's an important detail. Now, what the hell is alpha glucose? Well, this is the difference between alpha glucose and beta glucose, right? So in alpha glucose, here on the left, H is on top and OH is at the bottom. And then in beta glucose, it's the opposite. That's the only difference. And this, unfortunately, you need to memorize, right? So just remember that both starch and glycogen are made of alpha glucose, the one here on the left. So, as promised, this is the summary. So here we have amylose, right, which is unbranched. So as I said, when a molecule is unbranched, right, polysaccharides, the glucoses are side by side, as you can see here, right? But that actually does not yield a straight line because there's an angle, right, to the bond. So it ends up having a spiral form, right? But as you can see, it's a continuous line. That's what it means unbranched, right? Amylopectin, on the other hand, as you can see, they're still side by side, the 1 to 4 bonds, but then there's also 1 to 6, and you can see that here, right? 1 to 4, and then on top of each other. And it leads to this sort of tree shape, right? And then finally, glycogen is even more branched than amylopectin. That's a good thing to know, right? So it's also branched, but it's actually even more branched. So basically, to summarize, these polysaccharides, right, are very large molecules. As you can see, there's many glucoses. There can be tens of thousands of glucoses. And this means they're way less soluble, right? So they don't dissolve in water as well because they're so big. But this is amazing because what would happen if we had these humongous structures dissolved in water inside the cell? They would cause osmosis to make water go into the cell, right? And the cell would swell and potentially even burst. So it's very good that they're insoluble, and also they're very compact, which is useful for storage, right? You get more energy in less space, so that's great. But as I mentioned, polysaccharides can also be structural, and that's cellulose. So cellulose is made of beta glucoses and is unbranched, so it only has these 1 to 4 glycosidic bonds. However, because it's beta glucose, there's an interesting characteristic, and it's that every other glucose is inverted in respect to the other. So as you can see here, the oxygen is up top, whereas here it's at the bottom. So it's been inverted by 180 degrees. This is to allow the OHs to be close enough to react. And this again is because it's beta glucose and not alpha glucose. What this means is that actually cellulose is a straight chain. And therefore different molecules of cellulose can bundle up with high tensile strength. and they also have a lot of pressure resistance. Therefore, they form very solid structures. And here you can see it, right? So in the cellulose, you get these 1 to 4 bonds. So this is one molecule of cellulose, this is another, but they stack up to form these very, very solid structures. Whereas here, they're much less solid, right? But that's good because we want to be able to access it to break them and make energy. The next thing, this is a bit of an aside, right? But it's still important. It's glycoproteins. So glycoproteins are basically proteins that have a carbohydrate attached to them. Normally, it's an oligosaccharide, which is a short chain of monosaccharides, right? It's not a polysaccharide because it's not long enough, but it's bigger than a monosaccharide or a disaccharide. So these proteins are very often present in plasma membranes of cells, right? And the carbohydrate will be facing outwards. This basically allows other cells to recognize your cell through those glycoproteins. The glycoprotein can bind to a receptor on the other cell, right? And tell the cell something. What's a great example of this? The blood, right? So actually, you've obviously heard about group A, group B, group AB, group O. But what does this actually mean? This just has to do with glycoproteins. Basically, on our red blood cells, we can have 1 or 2 glycoproteins. So we can have A, we can have B, we can have A and B, or we can have O. But we can't have 3 at the same time, basically. This is very important for transfusions. Why? Well, I don't know if you've studied any immunology, right? But if you have, you know that your body rejects things that are not self, right? Things that are not recognized as yourself are rejected and killed. So basically what happens is if you're group A, right? And then you get injected with group B blood, your body's going to detect this glycoprotein because, again, as I said, it's cell recognition, right? And it's going to kill those cells. And that's not great. Same, if you're AB, right? You're going to everything is fine, basically, right? So if you are group AB and you get given any type of blood, you won't react against it. Because group O, interesting fact, is basically the same structure as A and B, but with 1 monosaccharide less at the end, right? Which means that AB does not recognize O as foreign, right? However, O recognizes everything else as foreign. Hopefully that makes sense. It's a bit complex, but that's the basis. Okay, and then moving on to lipids. So that's carbohydrates. Hopefully that gave you a good idea. Lipids are a bit more all over the place, I have to say. They're not as systematic. So lipids are hydrophobic. This is important. We said that glucose dissolves in water very easily. Lipids do not. They actually very rarely dissolve in water or any aqueous solvents. They do dissolve in non-polar substances, right? The main types are fats, oils, waxes, and steroids. And here are some things where you can find these in, right? So fats, lipids are not necessarily bad, right? A lot of them are good, but there's also bad lipids for your health. So again, similarly to what we saw before, how are lipids formed? Well, through condensation, right? The main types of lipids we're going to see in IB are triglycerides and phospholipids, okay? So basically, a triglyceride is formed from a glycerol and a fatty acid where there's 3 condensation reactions. So one here, and another one here. So 3 water molecules are generated, right? And then the final bond is called an ester bond. So a triglyceride is going to be the glycerol attached to 3 fatty acids. This R, basically, that you see here stands for a variable region, right? It can be of distinct lengths. But what can also happen is that instead of binding 3 fatty acids, it binds 2 fatty acids and a phosphate, right? So if it does that, you end up with a phospholipid, right? Where it's still the glycerol is here, then the 2 fatty acids, but then you get this phosphate, right? This phosphate head. What's interesting about this phospholipid is that it actually is partly hydrophobic and partly hydrophilic. So what I said before is not completely true. So we'll look at that in a sec. So as I said, triglycerides are completely hydrophobic, but phospholipids are partly hydrophobic, partly hydrophilic. That's called amphipathic. So when something is hydrophobic and hydrophilic, it's amphipathic. That's a key term to remember. So what does that mean? Phospholipids are actually found our plasma membranes are formed by phospholipids, the cell membrane, right? Why? Well, we have this hydrophilic head, so it attracts water, right? And then we have the hydrophobic tail, right? And we'll see this is normally represented like this, right? So these are the heads, these are the phosphate heads, and then these are the tails. So because the heads are hydrophilic, they like water, right? So they're going to like the external medium more. However, the tails are hydrophobic, so they attract each other more than they do the outside. This means that when you just leave them to be, right, phospholipids form a bilayer, right, a double layer like such, right? So they point towards each other. this is when you mix them with water. And as you remember, cells are mostly made out of water. So this is what's going to happen in a cell. Okay, so here you have it, right? We have this hydrophobic tail, or double tail in this case, and then the hydrophilic heads. And this is basically a plasma membrane, right? A cell membrane. This is how it's formed. It just forms naturally because of the characteristics of phospholipids. And then we have triglycerides, right? Well, fatty acids are actually in triglycerides and phospholipids, but when we talk about fatty acids, there's many differences. They can be saturated, right? You saw before that fatty acids had an R, right? This R was there because normally they're really, really long, so you don't want to draw the whole thing. But basically, they can be either saturated, where basically every carbon is filled with hydrogen, yeah? And then they can be unsaturated, where there's a double bond or more. So if there's one double bond, they're called monounsaturated, and if there's more than one double bond, it's polyunsaturated, right? So that's important because it determines its shape. So within the unsaturated ones as well, this is quite important to understand. You can have cis unsaturated, where the hydrogens are on the same side of the double bond, or trans, where they're on opposite sides of the double bond. If hopefully you can see that clearly. What are the big differences between all of this? Well, this has an impact on the melting points of these structures, right? Saturated fatty acids, as you can see, are pretty straight, right? They're a pretty straight chain, so they're going to pack very nicely, kind of like the way we saw in cellulose, right? A straight chain forms bundles with others. So this is going to pack very nicely, and it's going to be quite hard to remove. That's why it has so saturated fats have a high melting point. Unsaturated fats vary. So cis, actually, this type of bond induces a bend, as you can see. And this bend means that actually they don't stack up as well, right? Because they're not as neatly shaped to form bundles. So cis fatty acids, cis unsaturated fatty acids, are going to have a much lower melting point. Whereas trans fatty acids, because the hydrogens are on both sides, they form very straight chains again, meaning that, again, they pack very tightly and they're very hard to remove. This is why, in general, and this is a generalization, but saturated fatty acids and trans fatty acids are unhealthier than cis fatty acids, right? Because the cis fatty acids are easier to get rid of. So examples of a saturated, and this makes sense, right? A saturated fatty acid is butter, a cis unsaturated is olive oil, and a trans unsaturated is margarine. So actually, a lot of times what they do is they take vegetable oils, and because they want to make it solid, they partially hydrogenate it, making it trans, and that's actually very unhealthy, and it's been banned in several countries. So definitely avoid that in your next supermarket shop. All right, so this leads me on to triglycerides. So we said that phospholipids, their function is basically in cells, right? They form the cell membrane. That's a really important function. But what's the function of triglycerides? Well, here you have these really cute penguins, which illustrate the point. And it's that triglycerides are firstly used for long-term energy storage. So carbohydrates are short-term energy storage, right? The minute we need energy, you normally start using those carbohydrates. But afterwards, if there's no carbohydrates, you will use energy storage. So that's stored in adipose tissue, right? The triglycerides. But also triglycerides, right, are very chemically stable. They do not mix in water as well, right? And they have twice as much energy per gram as carbohydrate, right? So actually this makes them very good for this long-term storage, right? Because they're very stable, they're going to last for a really long time, and if you need them, they're going to give you a lot of energy. However, another very interesting function of triglycerides is that they're a very poor conductor of heat. This means they give you thermal insulation. And also, they're very liquid at body temperature, so they act as a shock absorber. And that's why you see a lot of animals that live in very cold climates, they have a lot of adipose tissue, right? It's because it acts as thermal insulation. So in the case of these penguins, right, I'm sure the triglycerides are giving some energy, but it's mostly because they protect them from the cold. And then finally, we have steroids, right, which we briefly mentioned before as one of the types of lipids. You don't need to know much about these, just know how to recognize them. They always come in the same forms, right? They're basically fused rings of carbon atoms, right? So rings, but you see they're different to carbohydrates, right? They're fused to each other. So they're mostly hydrophobic, and so they can just pass phospholipid bilayers. Basically, they can enter and leave cells very easily. Two examples are testosterone and estradiol. So these can be found naturally, but also these are the steroids that a lot of people take, right, to grow muscle, etc. Great. So now that's the end of the content, and now let's do some questions to recap. So first, which of the following polysaccharides are made of alpha glucose monomers? As always, I'll give you 3 seconds to think, and in three, two, and one, and you can pause here, of course. It's glycogen and starch, right? Remember, cellulose is structural, and it's formed of beta glucose, whereas glycogen and starch are energy storage, and they're made of alpha glucose. Great. Then, which of the following bonds links two monosaccharides together? Now, this is a bit more niche. You might not remember this one. And this is the one I put at the start. So let's see if you've figured it out now. In three, two, and one, it's a glycosidic bond, right? Ester bonds are in triglycerides, hydrogen bonds are in water, peptide bonds are in proteins. But glycosidic bonds are what form between two monosaccharides. Then, what is a characteristic of lipids? Again, this is pretty obvious, I think. Three, two, and one. They provide insulation. So they're not rapid energy release. That's carbohydrates. They're not soluble in water whatsoever. And they're not made by peptide bonds. That's proteins, right? But they do provide thermal insulation. Great. And last but not least, the image shows a fatty acid. What type of fatty acid is this? You should be able to do this. Pretty straightforward, I think. Again, pause to think. But in three, two, and one. It's polyunsaturated and it's cis. So unsaturated because it has a double bond, polyunsaturated because it has two double bonds, right? And cis because on both double bonds, you have the hydrogens being on the same side of the double bond instead of an H here and an H here, for example, right? Where it would be trans. So that's the end of this chapter. Sorry, it was a bit longer than last time's. And next week, we'll be back for B1.2. So if you have any questions, leave them in the comments. And until then, have a great week.